Hundreds of thousands of researchers around the world are working to improve life and address imminent threats to humanity. Often their research ends up in the scientific valley of death in the form of publications and patents that never see the light of day. That is about to change. Welcome to the Lab to Startup podcast, hosted by Naresh Sunkara, founder and executive director of the Berkeley Postdoc Entrepreneurship Program at the University of California, Berkeley. This show has two main goals: share the stories of those who have successfully founded startups based on their own research, and highlight resources needed to help those aspiring to launch startups in the deep tech space. Whether it's electric cars, vaccines, addressing climate change concerns, or possibly establishing life on other planets, Naresh and his esteemed guests want to help scientists, engineers, faculty, and researchers bring their innovations to market. Learn more and subscribe today at labtostartup.com. And now, here's Naresh.
My guests today are Professor Adam Abate and Charlie Silver, two of the co-founders of Mission Bio. Mission Bio is a startup that pioneered high-throughput single-cell sequencing, which is helping making precision medicine a reality for cancer patients by assisting in cancer research and biomarker development. Adam is an assistant professor in the Department of Bioengineering and Therapeutic Sciences at the University of California, San Francisco. His research interests are in the high-throughput biology with microfluidics, protein engineering, and biophysics. He received his PhD in physics from the University of Pennsylvania and a postdoc from Harvard University. Adam is a very special guest who is one of the very few faculty members that I know of who asks himself and his trainees a simple but powerful question: why am I doing this research? Other than getting a research grant or a job or a publication, how can my research be useful to the taxpayers funding my research? And Charlie is the founding CEO of Mission Bio and has been a very good friend of mine for a long time. He got his graduate degree in astrophysics from the University of Wisconsin and an MBA from Columbia University. Charlie has experience in product development along with marketing and business development before joining Mission Bio. Charlie is a deep thinker and a successful leader who played a crucial role in building Mission Bio. In this episode, we will learn about Adam's guiding principles for his research lab, founding story of Mission Bio, technology behind the product, fundraising. They've raised about $125 million since founding, partnerships they built along the way, and how competition and customers shape their product development, and leadership transition as the startup continues to grow. I'm sure there's a lot to learn from Adam and Charlie, and I hope you'll enjoy my conversation. Adam and Charlie, welcome to the show. Adam, I was doing some research on you, and what stood out immediately was your intentionality as a professor to build technologies that are useful to people and not just indulging in research in an academic lab that satisfies your intellectual curiosity, which is very commendable. And this is important to me because it is one of the themes and goals for this podcast to help faculty and trainees to think about launching startups. Would you like to elaborate on this topic, please?
Yeah, sure. I guess I have some strong opinions about it. This basically comes from, essentially, it's a part of my personality is that I'm pretty good at thinking of complicated ideas and things. I did my degrees in physics. I really like the deep conceptual aspect of research, but I also just have an inherently practical side to my brain. I'm always thinking about what's the value, what's the utility, why am I doing this? And I think it explains a lot of why I'm at UCSF. I was trained in physics. My PhD was in condensed matter physics. My postdoc was in physics. My undergraduate was in physics. So how did I end up as a professor at UCSF, which is Paramount Biomedical Research Institution? And it really comes from that realization that I'm practical by nature, and I want my work to have a practical output. I want it to be more than just fundamental science. And if you come from the engineering sciences or physical sciences, it's difficult to discover problems that matter when you're in that vacuum full of other quantitative scientists, physicists, and so on. All those people tend to think about the same things, but you really have to push yourself outside of that comfort zone in order to be exposed to the problems that actually matter. And that's really why I came to UCSF. It's an environment where the problems come to you. You're just immersed in an environment of problems that need solutions. And if you're pretty ingenious about how you solve problems and you can really bring a complementary skill set that doesn't already exist in that environment, then I think there's a lot of opportunity. So I think that's really a lot of it, is that I've built my lab with the mindset that we're going to leverage our fundamental skills in quantitative thinking and physics, but we want to apply that to solving biological problems. And so I always ask my students, actually on a regular basis, they're probably sick of it. I always ask them, take a step back and tell me why you're doing this. What's the point of this project? It always amazes me how many times I have to repeat that question until I actually get the answer I'm after, which is the motivation for why you're doing it. But don't tell me how it works. Don't tell me how it's better than the other technology. Tell me why you're spending your life, 5 years of your existence, working on this thing. What's the point? And I think so few scientists at this career stage, at the graduate level, and even at postdoc level, have taken the time to just think about that. Why am I doing this? What do I want out of my career? You always are just thinking about the goal of, I want to ultimately be a professor. I want to graduate. And these are really intermediate goals, not existential goals. And I think that's really the key if you want to be successful in commercialization, is that you need to solve big problems that matter to a lot of people that are beyond you. And if you're not asking yourself that question of, why am I doing this? Why does this matter? What could be the potential impact? You're just never going to do that. So I think that's kind of the key to it.
Very important question to ask. I think that I struggle with this, and I keep asking this at Berkeley and other places too. Ask yourself, please. I think that is important. Like, it's beyond your publications and your graduation. A degree doesn't mean much. I keep talking about this. All the technologies end up in the scientific value of death in the form of publications and patents. Who's going to take them and do something about it? But I'm so glad you're doing this. I think this idea of questioning yourself needs to be propagated more. On that note, Charlie, I'll turn to you. In one of the interviews, you mentioned about how only about 3% of the R&D pipeline is making to the market in the form of drugs. We are in 2021. We spend about $100 billion around the world in drug discovery. It's a very bad number. How, in your vision, is Mission Bio helping change that statistic?
Yeah, this has been always one of the guiding principles behind Mission Bio as a company, this mindset that drug development, which is for good reason, sucking out a lot of the investor dollars from our ecosystem and, of course, taxpayer dollars through NIH. A lot of this drug development money goes to very important underlying biology, but all too often these drugs fail along the way. So you end up with really strong preclinical assets that fall down when they get into patients. And it's nothing new. I think, if anything, what's new is a recognition that precision drugs, when they work spectacularly. And there's real reason to continue investing in these, because when it works, it works. And we've seen that the patient need is there. And when technologies come to bear, it's unquestionably worth it to be making these investments in drug development. But the numbers, the failure rate, the low success rates of these drugs actually making their way through to patients, I think opens up the way for more precise measurement technologies to support the entire drug development pipeline. And that's always been one of the guiding principles of Mission Bio, this mindset that the technologies we are using to study biopsies, the technologies we're using for basic underlying biological research are blunt hammers and very imprecise tools. And that there's really room for precision, in the case of Mission Bio, for precision in sequencing to help understand every little bit of insight that every cell has to tell you about the disease. And the guiding principle is that hopefully, if you have all of the tools available to you to understand what the disease is doing in the patient, how the patient is evading the disease, and why, and what's the underlying biological mechanism. Once you finally have that precision toolbox to be able to deeply understand what's happening to the patient, then you have a shot at really making many more of these drugs successful. And I think every dollar that is spent in drug development in this country is extremely highly leveraged, because we're always learning from our failures and we're always doing better. So I'm always hopeful that Mission Bio as a company, by continuing to provide these precision tool sets across the entire chain from preclinical through to on-market biomarkers, can really help improve success rates for these drugs and help develop medicines for the patients who need them on a faster timeline.
Great. Thank you for that. The reason I asked you both those questions is because at a higher level, the mission of both of you, the co-founders, aligns in terms of the big picture. It's a very important characteristic that I found about successful co-founders, being aligned in your individual missions as well. So I just want to put that point out for the people listening to this. So let's just dive into this subject. We are talking about Mission Bio. Adam, for the sake of audience, please describe the problem you're solving at Mission Bio and the research at your academic lab that led you to think about launching a startup. What is that aha moment or the decisive moment that you determined, I need to start a company?
I think honestly, when it comes to technologies that make a big impact, hindsight is 2020, and looking forward is almost impossible. I think it's the nature of technology. It proceeds in leaps and bounds, and it's almost impossible to predict which is going to have the big impact until it's already happened. Sort of everyone sees it at the same time. And I think when you get to make one of these leaps as a scientist, it's because you happen to find yourself in a unique set of circumstances where, for some reason, you have that ability to see the next big step forward. And for me, it was really circumstantial. I mean, it happened that I was doing my postdoctoral work in a lab at Harvard. The lab of David Waits is a very famous physicist, a very successful entrepreneur himself. And that lab specialized in building these fairly intricate devices for doing biological assays. Quite honestly, at that time, we didn't really know what to do with it. We just knew it was a cool technology. And I think this is also true of a lot of technologies that end up being groundbreaking, is when they're being initially developed, no one quite knows what they're going to be good for until they connect to that problem. Basically, when I came to UCSF, I started looking around at what a lot of people were really interested in. And one of those areas, kind of generally speaking, is high throughput screening, where they wanted to do a lot of assays, usually genetic-based assays, where you want to interrogate a large library of DNA combinations. So you'll have DNA, you'll mutate it, and you want to interrogate all of these sequences. And the cost of doing that is really, how many assays can you do reliably? And if you want to do hundreds, yeah, there's ways to do that. But if you want to do millions or hundreds of millions, it's well beyond the realm of possibility. And that was exactly what I was trying to build my technology to do as a postdoc. And when I came here to UCSF and I was thinking about applications, it occurred to me that a similar thing could be useful for doing sequencing, because in many ways, doing a sequencing assay is not very different from doing one of these high throughput screens. Ultimately, you have a bunch of test tubes and you want to analyze the contents of each test tube. And when you're doing sequencing, your test tubes contain single cells and you want to analyze each individual cell. And as soon as I reduced the problem down to that kind of objective, what I realized is that it would be quite straightforward to do that. Technically speaking, it wasn't that difficult to actually build the technology once I'd had that idea of the technology was going to do, which is this very high throughput single cell sequencing. The next challenge really became, well, why in the world would anyone want to sequence, thousands of single cells or tens of thousands of single cells? And that's really where I think we started the real struggle as a company, was to really, at that time, because you have to remember now, single cell genomics is everywhere. It's an enormous field, huge amount of value. But back in 2011, 2012, I had grant after grant rejected saying, what are you going to do with this? What problem are you going to study? Yeah, that would probably be, I think, how it came about. And I guess what I would say is that it wasn't like I had this clear vision, like, okay, we're going to use this to do personalized cancer treatment, to make selecting the treatments that are going to actually cure disease as opposed to delaying the inevitable. That wasn't the goal at the beginning. At the beginning was really this realization that we had a technology that had the potential to do something really cool sounding, sequencing tens of thousands of cells. And then really with Charlie's help and doing a huge amount of discussion with people in the commercial sector and other biologists, that's when we really hit upon the cancer application and how this could actually make an impact to actually people's treatments.
So this was 2014 you launched the company?
I started the company formally, I think, in 2011 or 2012, but it was really just a placeholder. At the time, it actually was called Torrent Bio. It wasn't even called Mission Bio, because I knew that there was something to the technology. But the company didn't really get off the ground until Charlie and my other co-founders, Adam and Dennis, got involved and took my very early idea of what this technology was and connected it to real applications. That is a market, or time no market exists for this, but the realization that there could ultimately be a huge market for doing these kinds of personalized cancer assays.
The reason I asked that question was because in 2011, they were not willing to fund these projects in the form of grants. I was trying to see if there were any tailwinds that changed your decision to launch a company. We'll come back to that, but walk me through this conversation with your co-founders. There's Charlie, Adam, Dennis. What transpired that you decided that we should move forward full-fledgedly in forming a company?
So at the time, I had this idea of just starting my lab. I started my lab in early 2011 and basically had nobody in the lab, literally had nobody in the lab. So I was looking for super talented postdocs, and I knew I wanted to do something in commercialization because I had some experience as a postdoc. And so I was actively trying to recruit people that were interested in doing that. And so I went to a lot of these mixer events and so on at UCSF, where the goal is to bring together a bunch of people that are all interested in trying to start a company. And that's how I got connected to Adam Chiambi, who was a co-founder of Mission Bio, and Dennis Eastburn. Adam was a recent graduate of Stanford Applied Physics, and Dennis had been a postdoc at UCSF in a cancer biology lab. And I told, hey, I want to create a company. They said, hey, that's great. I want to create a company. And so they came to work for me as postdocs. All this work was unfunded. It was supported basically by what's called my startup package, which is when you're a new professor, your department gives you just a bunch of money just to try a bunch of things out because they know that this is a problem where trying to convince the NIH that your idea is a good idea is really hard when you're totally unproven. You have no data and no history, basically. So we did all of this work with the money that was my startup. And Adam and Dennis, I kind of lucked out because they were spectacular postdocs. They were perfectly complementary. And we just started developing the basic idea, the basic hardware. And then that's right about the time where Charlie came into the picture because he was a business student at Haas and needed to do an internship. And he heard me talk somewhere, I don't remember where, and he's like, I want to do it with you. I think your idea is great. And I was flattered to hear that somebody with a business degree would to spend time on my crazy idea. It was really that combination because to start a company, you really need the whole range. You certainly need technology and that scientific expertise, but even more important, I would argue, is the commercial expertise. And that's really what Charlie brought on board. I think that was around 2012, 2013, maybe Charlie remembers better than me. And we basically worked really hard with no money to just evolve this idea to the point where we realized there was an opportunity here to actually raise investment capital. And this was still before a lot of the companies in this space had emerged. So we were very early, but it was also clearly riding an uptick where there was clearly going to be a lot more interest in companies that could leverage sequencing for precision genomics. That's kind of how that took off.
Interesting. Unlike a number of other professors who start their career, not many look into commercializing technologies and they just want to get that tenure, go down that path, but you made this choice. Charlie, walk me through the decision process. When you talked to Adam and the rest of the team, what was that deciding point where you said, like, I want to join this team and want to work with these guys? What are the factors you're looking for in a co-founder at that point?
Sure. When I came into, I was going to say Mission Bio, but it was Torrent Bio at the time, Adam. Thanks for reminding me of that. When I came in to join up with this team, I was just wrapping up an executive MBA at actually the Berkeley Columbia Joint Business School. And in my very last semester of that program, I knew that I was going to be building my next one. And I took the time as I was working with Adam to sort through different technologies that I was connected to that were ready to spin out from a university. So it was actually a very deliberate and thoughtful approach to look at technologies with IP and people attached to them that were ready to commercialize with a very kind of systematic approach to working up each of them. And one of those was Mission Bio or Torrent Bio at the time with Adam and the team. And what I was looking for was really the founding team. So it was number one, a technology that had platform potential with the ability to move in a lot of directions wherever the market might want to take it. I was looking for a technology that would really have great impact, particularly impact in health, although I was looking across industries at the time. And above all, the founding team was the most important piece to me, a technology that was mature enough to be able to pull out together with the team around it that had developed it. Because to me, that team, the initial momentum always comes from the founding team and those first key employees. And to me, it was really important that we had people who were able to immediately come out and staff the company with the technology that they had already built on the bench. So the IP is not enough. It's not enough just to have technology working in the lab, but the people to continue commercializing it to me was just essential. And out of all of the technologies that I sorted through, the team that Adam had pulled into his lab with a very thoughtful intention of spinning out a company around us, I recognized as a mature enough team that we could very quickly come together and build something as soon as it was off the bench and into industry. So that readiness for market and the readiness of the team to build it to me was the number one thing. I recognized the innovative capacity of Adam himself, as well as the innovative and just sheer energy of Adam Shiambi and Dennis Eastburn, who really complemented what Adam brought from the academic side with expertise in just building rapidly and then applying that to important applications in molecular biology, which was what Dennis brought in. So I saw that the three of them, together with the background that I brought from my own prior experience in product development, really completed what we needed in the early days for the team, which was just head and shoulders above other opportunities that I was looking at. It was really that team capability that brought us together.
Fascinating. So here you are, a passionate group of people a fantastic technology that you all believe that there's some meat in there that you can commercialize. The next step, how do you decide on the roles? Adam, I believe you're going to be still a full-time faculty at the university. Walk me through the process of like the deliberations in deciding who's going to do what at the company.
Yeah, I mean, in many regards, I think we kind of just naturally fell into the roles. I knew I wasn't going to leave my post as a professor at UCSF, so that was pretty easy. I was kind of the academic founder and board of directors and very closely collaborating with the company, but I wasn't going to have a formal role. Charlie was the clear CEO. I mean, he was the only person that had business experience, and he'd already been very successful focusing the vision to actual markets. And in the end, was able to raise a huge amount of venture capital. So at that time, when we had to make the decision to make Charlie CEO, he hadn't done that yet. But it was clear just talking to Charlie and hearing about people he was talking to, the network he had, that he could be successful. I believed in him. And basically that was why I supported that. And then Dennis and Adam, they're scientists fundamentally. So they were happy to take scientific roles at the company. So there was no real deliberations, who's going to do what, or any kind of disagreements about that. It was really just, we all have our talents. We know what those talents are, and we know what we're not good at. And because we had this complementary team from the outset, the roles were very natural.
And one conundrum a lot of faculty I've seen is like, how do you demarcate yourself in terms of your involvement? There's a full-time CEO, but then the technology is coming from your lab. How much did you get involved in business decisions? What was that looking like in the beginning?
Yeah, I mean, I talked to Charlie about this very early on. And actually, to be totally honest, one of my concerns was that Charlie, Dennis, and Adam would think that I wanted to have control over this company, because I recognized from the beginning that I was not qualified to start a company, at least by myself. I needed a team, a complementary team. And so my role at Mission Bio and what I tried to really facilitate was, how can I help Mission Bio? Call on me as much and as often as you need, because I'm here to help. I want to do whatever I can to make it a success, but I'm just one voice. I'm not going to be the determining factor. And in the end, it became very clear then how I would interact with the company, because the reality is that I am the CEO of essentially my lab, right? That's my company, is my lab. And Mission Bio is my offspring to some way, but I was not the CEO of that company. So really it was a partner. And so it was a very clear partition for me in my mind, where my responsibility lies kind of as the CEO, the decision maker. It's with my lab and not with Mission Bio. But I always asked to be part of big decisions, to as much as possible be involved in discussions, even recognizing that I was not actually formally an employee of the company. And Charlie did a really great job of keeping me in the loop. He didn't have to be clear. I think he did it because he saw value in hearing my voice, and that he knew that we could work well together, and that could be really hugely important to the success of the company. I think there's actually a lesson I took from this, which is a more broad lesson about commercialization in general. I think a lot of people who haven't created companies before tend to play hardball in their negotiations with the people they're creating the company with, how much equity they're getting, what their roles are going to be, how much control they can maintain. And I really think that's a mistake, because it's short-sighted. At the end of the day, you guys are all on the same team and you're trying to create something out of nothing. And what that means is that you're shooting yourself in the foot by having these hard-nosed negotiations with your co-founders. You just need to agree fundamentally on how things are going to work out. Because if you don't have that agreement and you have a difficult founding situation, the company's never going to work. There's too many challenges to ultimately make it work.
Thank you so much on those insights. That clarity is so important. The reason I actually asked that question was like, I wanted to give that advice to other faculty members considering those spinouts. Like you said, some people try to play that hardball. It doesn't matter at the end of the day, I think, if you don't put your hearts together as a team, the company is going to fail. Any thoughts from you, Charlie?
That's one of the most important lessons that, if I could be just really specific in giving advice, that's one of the most important pieces of advice I give early stage founders, and particularly first time academic founders, as Adam said, because we've lived through this by sheer experience. And that is that the numbers just don't matter that much. It doesn't matter if you've got 19% or 20%. And I say the same thing to investors, because at early stage ventures, I think there's a temptation for people to overanalyze the risk reward ratio. And I think that's just as true for investors, if not more so, as it is for first-time founders, and particularly academic founders, because they tend to be just extremely analytical to their detriment in that negotiation. And really what I mean by that is that if you've got 19%, if you've got 20%, 25%, it just doesn't matter that much. What matters is the success of the whole venture. And what matters to that is that the entire venture views it as a partnership, not necessarily of equals, but of equal contribution. In other words, that everybody is pulling together in the same way to make this venture successful. And if everyone is pointed forward together, if you really view it as a partnership, much more than kind of startup building, if you view it as a team and a partnership and a collaborative effort between entrepreneurs, founders, management, company, and investors, everyone is pulling to make it a success. And if it's a success, the numbers don't matter that much. They don't matter in terms of what the company does on the market, and they don't matter that much in terms of an outcome for the company, because everyone will be successful within certain boundaries. Everyone will be successful. In addition to entrepreneurs and founders, there's often a temptation for the lawyers who advise early stage ventures to spend a lot of time arguing about the boundary cases, which is what lawyers do. But again, to the detriment of the long run success of the venture, what really matters is that you pull together a team that's all going to pull together to make this thing a success. If it fails, everyone fails together. And if it's a success, then everyone wins. And it's just so much more important to build those long-term collaborative relationships than it is to get to arbitrary precision on the numbers. And from the investor side, the earlier you go, particularly for these angel rounds, even for Series A, the outcome is so wildly unpredictable in the early days of a venture. You know, in some cases, you've got to really put your finger to the wind and assume that this founding team, whatever it takes, is going to get there in some way or another with some technology they'll pull into it. And it's really hard to model out that sort of thing down to super fine precision. So at some point, you make the plunge and you jump into it. I very much echo the comment that Adam made. The numbers matter much less than the relationships around the table.
Great points. Great points. Those relations really matter as you build a company. We'll slightly shift the gears. I wanted to get into the product side of it, like the Tapestry platform. Charlie, if you can give a quick summary of like what Tapestry is and like give a good example, a quick one, like from sample collection to results. What happens in that platform?
Sure thing. So Tapestry is the market leader for single cell genomics on the translational and clinical side of the space. And what that means is it's adopted by most of the cancer centers in this country, many of the medical research institutes worldwide, and many of the leading drug companies worldwide to help them accelerate their drug pipeline. And they do that with the precision that Tapestry provides by analyzing the genomics of every single cell at the resolution of the cell, which for the applications that Mission Bio supports, the cell is the resolution that matters. So we provide the fundamental resolution where biology operates. And what that enables our partners to do is to really understand at the level of the cell how cancer biopsies are evolving or escaping therapeutic resistance, how edited cells have either been properly edited or inadvertently edited, and in general, how any single cell biology application can really have impact in translational and ultimately in clinical type applications. So very specifically, what the platform does is it analyzes the DNA of every single cell in a sample. Recently, Tapestry added capability for looking at DNA and protein simultaneously. So it's the first time ever that the market's had the capability for fundamental ground truth resolution by linking together the genomics and the functional result of those genomics at the resolution of the cell, which is where it matters for patient care. And the platform is delivered as an instrument consumable product suite. So the instrument is what we call the Tapestry. The consumable underneath are reagent kits that are used to add barcodes to every cell in the sample. And together with adding the barcodes, amplify up gene-specific amplicons against the cellular genomics as a way of understanding what's the DNA content, and side by side with that, tagging the proteome to understand, well, not across the whole proteome, but for certain protein markers, how does the protein talk to the genomics down to the level of single cells. So the product suite to do that is an instrument that runs the assay kits, which are microfluidic cartridges together with the reagents. So it's a full product suite, including the software at the back end, which tends to be very important for customers, especially in the translational space, because having that full suite of analytical capability through to the insights that are generated is just really important for enabling applications for studying patient populations. So it's provided as an instrument consumable set with the software at the back end, and it's used for applications for studying therapeutic resistance, where there are known genomic markers that are tracking the evolution of disease in response to therapeutics to understand what every cell is doing or how the cells are not responding to therapeutics as a way of getting the most precise measurement of what that biopsy is trying to tell you.
Fascinating. Let us go back, 2011, I guess. What was the hypothesis when you first started the company, Adam and Charlie? What was the vision for the product when you first started, and where is it today? And how did that evolve over a period of time?
I'll just say a little bit and then let Charlie take it over. But I'd say it evolved enormously. From my perspective, being really a technology developer, tool developer, I just thought it'd be cool if we could sequence the genomes of thousands and thousands of cells routinely. And we started out with different ideas about how that could be used, but it really took a few years to evolve into this idea that it'd be hugely important for precision cancer therapeutics. So, Charlie, do you want to add anything to that?
Yeah, I'll say we went through many evolutions in terms of capability of the technology. And I wouldn't really call it options, because over time, we developed this foundational capability that Adam and Dennis had developed in the lab. We developed that into many different product configurations that could ultimately be commercialized. So I would say in the early days, when we launched the company as a spin out from UCSF, we knew we had a platform, and we knew this platform had enormous applicability across many areas of basic biology and many disease areas impacting human health. And we didn't exactly have a specific idea of where the product was initially going to pull in the market. And this is, way back to 2014, as we were still pulling the venture together, because we did evolve into it pretty quickly. But that platform capability, I think, was so important because in the early years of the company, we really funded it off government grants side by side with service contracts for industry. So a whole collection of non-dilutive capital really funded the early days of the company. And by doing that on top of the platform, we were able to develop every single product configuration that we could ever build, at least in the early days, and many that still are very much there in a way that we could build it when the time comes. And these were not just ideas, they were publications. In many cases, they were publications with important collaborators from Adam at UCSF or from across the ecosystem with real applications behind it, not just basic technology demonstration. So with the basic underlying platform that Adam and the team developed, in the early days, we demonstrated, and again, all of these are published, we demonstrated what we called PACS, or PCR activated cell sorting, which is a way of sorting the genomic contents of cells based on PCR fluorescence. So think FACS for flow cytometry, but instead of sorting off antibodies, you're sorting off true PCR fluorescence. We developed a qPCR assay, so a very, very high throughput multiplexed qPCR readout. We developed the ability to sort the genomic content of free DNA. So even aside from the cells, just free DNA circulating in the bloodstream, we developed a way to very specifically pull down and sort DNA fragments and then sequence them downstream. And of course, we developed the capability for single cell DNA sequencing, as well as many other configurations of the technology. So in the early days, that non-dilutive capital allowed us the luxury of playing around with a lot of different product configurations and demonstrating that the platform has legs. And we were able to do that hand in hand with these industry contracts that provided real world validation on different versions of the product, so that when we finally ended up identifying where the market wanted us to launch initially, we had a whole toolkit to pick from. And the company still very much stands on the foundation of that initial toolkit that we had built out in the early years. And that toolkit on top of the fundamental technology was really a long run strength of the company. So in terms of identifying the first product to build, we were able to canvas the market as we were deciding which application to launch, which market segment to launch into. We were able to canvas the market and come back and say, not just, yeah, you can build this, maybe you should build this, but we had an entire suite of technology that we could build. And we were able to draw a very sharp circle around what customers were telling us and why we should build it as the first product. And because we had those different configurations of the technology already validated, we were able to move the technology through to market in the first product very quickly, which was what turned out to be that initial single cell DNA assay with applications in therapy resistance for leukemia. But because of all of that early technology development, hand in hand with the Abate lab, even after we spun out, we were really able to move with the market very quickly.
I'll just add a little to that too, that, early on, we also did have some really difficult decisions to make that locked us in, that still today, we don't know if it was the right decision or not. So for example, at the time, single cell RNA-seq, which has become a hugely valuable and important area today. I mean, there's companies valuing in the tens of billions of dollars just on that one product. Now, that was something that didn't exist yet, or I should say there was one company called Fluidigm that had some very primitive technology in that space, with very limited in scalability. And we actually could do much, much better with our technology. And so the founders, we actually discussed, should we be building a single cell RNA-seq technology, or should we go towards a DNA-seq? And the decision we made at the time was basically based on competition. We said, well, actually, the RNA-seq technology, it turns out, didn't require anything that didn't already exist. The technology that predates Mission Bio and the technology in my lab is used in those RNA-seq technologies. And so what we said, well, this means that there's going to be a lot of competition there. And so we actually decided to walk away from that product, basically on the hypothesis that we wanted to build a unique beachhead where only we would be able to play, only our technology would play, it would be supported by our intellectual property, even though we recognized at the time that that was still a much less proven market than single cell RNA-seq. That was a decision we made, and that has actually in many ways set our fate to where we are today. But I still sometimes think about, what did we decided, Charlie, to double or triple down just on RNA-seq back in 2013? We could have. And who knows where Mission Bio would be today had we done that? We'll never know. We could have failed because we could have lost to the competition, or we could have been hugely successful.
We'll never know for sure, but I actually think, Adam, that decision sort of led to one of the key strengths of the company, which is the fact that we did the hard thing. The market inevitably started with the easy thing. Fluidigm had already played around with single cell RNA. 10x was already looking at that market and starting to build into it. And RNA is easy. You pop open the cell and the RNA is right there. DNA is the hard thing. And we knew that DNA is inevitably going to be more impactful for translational and clinical medicine, which is always the way genomics has played out. RNA tends to be a research analyte. DNA tends to be where the clinic ends up for a lot of different reasons. But we saw that playing out. And we were able to do the hard thing, I think, Adam, because of the partnership with your lab and because of the innovative capacity of you and your entire lab, including Adam and Dennis, who came out with the company. I think that willingness to take bold new ideas and turn them into bold new technology on the market has proved to be a real strength of Mission Bio, because we did the hard thing. We did the thing that needed to be done because the market needed single cell DNA. And this really is an important side of the market in terms of applicability to real benefit for patients. But it took a hard technology to be able to get there. And I think doubling down on what's hard with the innovative capacity out of your lab has turned out to be a competitive advantage, because, again, it's hard to do. Others have fallen down trying to get there, but there's a real reason to do it because it's an important side of the market. So I think, for better or worse, it's actually turned into a strength for Mission Bio.
That's a nice discussion there. Competition and customers seem to shape the products of successful companies mostly. You might win some, you might lose some, but looks like you guys are going down the right direction. You talked about industry partners and academic partners. Was there like one or two consequential relations along this journey that actually shaped some good products at your end?
Absolutely. When we first spun the company out of UCS, we had government grants to get it underway, but those were fairly small, and that was what got us up and running in the incubator. The large contract that really got the company launched was a partnership with J&J doing circulating tumor cell analysis. So this was at a time when J&J was developing their next generation CTC isolation. We partnered up with them as a way of detecting the molecular analysis at the back end of every CTC that they were able to pull out of a patient sample. These are circulating tumor cells that are shed by the tumor and can be detected in very, very low concentrations, like one cell or several cells per milliliter of blood draw. So extremely rare cells, but extremely informative. And by plugging the two technologies together, we were able to come up with something that the market really hasn't seen before, or really even since then, and came away with some very, very important results together. That was a large joint development contract that funded the early days of the company, but really moved us from research toward real world applications, because that really pulled the technology into a direction where it had to run routinely, reliably, with high quality, with good repeatability across many patient samples. And that is no small hurdle to ask of a new technology that is just spun out of a university. So that industry contract, I think, by forcing us to run through some of the most challenging real world samples out there, just served to leapfrog the development of the technology. So when we did end up finally building the product for single cell DNA sequencing, we had all that learning behind us. And a lot of the important infrastructure considerations around the platform technology: how do you transfer the samples? How do you store the samples? How do you get the cell into the microfluidics, into the platform with the right quality and the right cell quality? So we were able to shake through all of those before we even started building the product. And that brought us a long way. I would also say from the early days of the company, there's been a real mindset that continues to this day that it's really important to partner with the thought leadership across the space, both academic and industry thought leadership, as a way to bring credibility to a new technology and to demonstrate to the market all of the important applications that can be accessed with a new technology. And right from the early days, we had just absolutely key partners across UCSF, MD Anderson, Sloan Kettering, the Hutchinson Cancer Center, Stanford, and others like them that really helped validate the technology, run real world samples, develop applications, and publish on it to show the world what could be done. And I'd say all of those were absolutely key partners to the company.
Yeah, this partnership seems to be playing a big role, especially in deep tech. You can't go by yourself and prove something without having those partners. How do you end up with partnerships like J&J when you have such an early stage technology?
I think it was the right match at the right time. J&J had a need for the analytics at the back end. They saw that coming. And we recognized circulating tumor cell analysis as a really important application for single cell genomics, especially for the single cell PCR assay that we had built out. So it was just the maturity of both technologies at the same time that made a lot of sense. And I think it speaks to the strength of the platform that right as we were spinning out, remember, this was a purely academic platform. Within 6 months of spinning out, we were able to close a contract of that scale and really start running real world patient samples on top of it. I think that just speaks to the strength of what Adam and the team had developed on the bench, which I think is another really important point for startups and spinouts from academia. It's easy to develop a technology, it's easy to get a publication out, but it's very, very hard to run it at scale and develop it into a form factor that the market will accept. So I think having a platform technology that's able to run in that setting so quickly after spinning out added a lot of credibility to what we did from there. I'd say one other thing around key partners, and actually this is an important story around how we've built Mission Bio. Right from day one, and I think ever since then, from when I first met Adam, I guess it was about 9 years ago at this point, I have been in the strongest way possible urging Adam and encouraging him to jump out with the stuff. In other words, the team around the table is always the very best one to build it. Adam himself has just tremendous innovative capacity that's not purely academic. It's extremely commercially oriented. And there is no one better to build the company than the folks who develop the technology. And I guess I have to offer an apology to the UCSF administration for encouraging that. But to Adam's credit, he's pushed back every step of the way, I think very successfully. But what that means is that we've actually leveraged Adam's lab at UCSF as, I would say, the most key partner to the company. And along the way, we've not just licensed out the technology and brought in the people from his lab, but we've also collaborated back and forth over the years. And the technology that we've developed, even the product that we built, went back to Adam's lab for him to continue building on top of. And Adam has continued to collaborate across the UCSF ecosystem with his collaborators on top of the foundational technology, which has really just served as a key sandbox for the company to continue that innovative capacity, even as we continue the commercial traction on the market. That's been, I'd say, one of the most important partnerships to the company from day one.
That is such an important point. Those relations with the lab at academia and then the startup working together is going to be so important. Keep that in mind. Keep good relations with the university and make sure both are benefiting from that. You talked about funding. Talk us to the story. Funding from having nothing to raising about $120 million. That's a lot of money. First of all, start with your story about how long did you go without a salary, and then segue into where you are right now.
I joined up with the team at UCSF. I guess it was 2013. I was thinking back at them, and we finally got the company spun out in 2014. So for that year or so, the company was still very much academic as we were getting the government grants in place to launch the company. So that was the first year of my involvement with the company. After 2014, we had those first grants in place. We had industry contracts beginning to come in, including the one I mentioned. And we really moved the company forward very quickly with a number of government grants. We ended up raising, before we raised our first dollar of venture capital, we had about $2.5 million in grants into the company. And by the time we raised the Series B, because we continued to execute grants, we were close to $5 million in grants and government contracts. And all of that went a long way to develop the early technology before the investors came in and added their dilution along with their capital, which meant that we were able to move the company really way past seed stage before we even raised the first seed dollar. So side by side with the grants, we started raising seed money. We raised actually a pretty substantial seed financing up until the Series A, which was in 2017 with Mayfield in the lead. We raised a Series B very quickly after that. So the Series A brought us on market very quickly, actually, after the Series A financing. And then the Series B got us to commercial scale. So the Series B was in 2018. We closed the Series C financing last year, which is really the growth round that continues to push the company into full scale. And along the way, among all of those venture financings, we've raised about $125 million to date.
Congratulations on that. That's a huge deal in the space. You mentioned about grants and contracts. Can you talk a bit more about those? Like, what kind of grants are these? What kind of contracts are these? How do you get them?
Sure. The grants were a combination of SBIRs and STTRs. Those are the industry academic partnership that's analogous to an SBIR, except that there's an academic involved. So we started just like many companies. We started with the phase one. I'd say just like many companies, Adam, you could probably remind us of this, but we didn't win the first one. There were a few cycles through, but we did fairly quickly, actually. We did end up winning that first phase one and piece in the second phase one behind it. We moved through to a number of phase two grants, both from the NIH and the NSF. And we also funded the company through a large government contract that was a consortium ultimately funded back through IARPA that involved upwards of a half dozen industry and academic partners, including both Mission Bio and UCSF. So all of those were non-dilutive government sources of funding along the way.
And I'd like to add a little something to that. I mean, let's be honest here. When you're starting a company, especially out of an academic lab, to a large extent, you see raising that seed or series as the endpoint. At least I did. Before I had done that myself, I had this naive perspective that, well, if I can just raise $20 million in Series A, everything else is going to take care of itself. And it's clearly a big milestone in any company to do that, but obviously it's not the end. The other thing I'll say, which is related to the government grant things, and actually to the J&J partnership, is that VCs, this isn't their first time to the rodeo. You're not the first company they're talking to at this stage. They've seen this many, many times. They've had extremely smart people pitch them literally on a daily basis about their idea. So how do you get over that barrier to actually ultimately close that private money? And I think actually, Charlie, I always thought that was kind of the biggest takeaway from J&J. I mean, clearly there was a lot of practical use in terms of how we understood the technology, how we ran it. But really, I think that it gave us credibility with the blue chip investors to take us seriously. And I kind of felt that after that point, there was a different tone in the room when we spoke with investors that we weren't just smart guys with an idea. They were starting to really think about writing a check. And that was not the case before that. We talked to a lot of potential investors and I felt like we just struggled to get anybody to really take us seriously until that happened. And I think where the grants come in is this issue, which is that how do you get taken seriously? Well, you got to build something that actually works. Okay. And something that works is very different from something that's publishable. I mean, from an engineering perspective, it's got to be something that you can go to a relatively independent person in that space and get them to say that they're willing to pay you for this technology. Now, if you think about any advanced technology that's in a lab, it doesn't look like it was built by a graduate student. It's pretty mature. It literally takes tens of millions of dollars to get to that point. And so you have a chicken and egg problem, which is that you need the money to build that mature product, but you can't get the money until you have the mature product. And a lot of the art of raising money, which Charlie was extremely successful with, was striking that balance. How do we get the technology up to a point where you could convince the veterans in this industry to write a check for you? And government grants are absolutely key to that process. I think it's called the Valley of Death. It's this idea that you have a technology that works in a lab, that works in the hands of your postdoc, and now you got to get it up to this point where someone's willing to give you their own money out of their savings account to take it to the next level. And government grants are basically the only way I know to do that.
As an academic, you're good at writing grants, and I think you should use that skill to definitely get some SPR or STTRs to kick off some development work. Charlie, can you give me a sense of how many investors you had to meet and pitch? I don't want to make it look very simple that you raised $125 million. I'm sure it was really hard work. Give a sense of like the timelines and what do you think convinced the investors that gave the money, and why do you think others could not invest in your company? What was the driving factor?
I've met every one of them and I've pitched to most of them. I think that's the short answer. And we ended up partnering with the ones that are the very best fit to the business. Adam's comment is dead on in terms of the validation provided by these early partnerships, both with the KOLs and also the industry partnerships and some of the pharma partners that we had early on as well. That's the credibility that you need to be able to talk to investors and convince them that this is the right next move for the market and this is the right technology to be able to serve it. So I think that really went a long way toward opening fundraising for us. And I think we stumbled into a lot of the right decisions early in the company in terms of providing that validation early on. In the early days of the company, we raised from a lot of angel investors who I still view as really, really close partners to the company because they've been close supporters to us from the very early days, who have not just supported initially, but continued to offer their support over time right up to today. So in terms of the earlier investors, we ended up raising from a lot of angel investors, which just brought a lot of really talented capital and early stage advisory in terms of support for the company. At the Series A stage, we talked to a lot of the life science investors. This was at a time a few years back when the market was just starting to change for life science investing. We had seen a lot of the funds that existed in the mid to late 2000s sort of shake out around biotools, where the few successes had played out and the predominant failures had played out as well. And we've seen a lot of these funds at the time back off on their investment in life science, back off on biotools. And frankly, some of these funds didn't end up raising follow-ons. So we were raising sort of in the aftermath of that, but also at the beginning of a trend for some of the bedrock Silicon Valley tech investors who are always at the leading edge of the very next wave, whatever the industry is. There's been a trend over the last few years where our Series A, I think, really aligned very nicely with the leading edge of this, for tech investors to start to take a close look at genomics and realize that Illumina sequencing is far and wide as a platform. And now there are opportunities for applications to go deep on top of the platform and really turn into what is now a pretty substantial market cap, both across the private and public space recently. So we were right at the beginning of that wave of the tech investors starting to take genomics seriously. And I think we really leveraged that in the early raises. So with Mayfield as an early partner, we brought in the strength of what Mayfield does as a fund, but with that sort of diversity among the investors that a tech investor naturally brings to a life science business. And it really served the company well from the early days. From there, we also brought in strategic partners. So in the Series B, we had Agilent invest, as well as LabCorp, which was another important strategic partner. Again, number one, to help build credibility, but number two, to also leverage their support, their expertise in carrying us forward into the translational markets that we were headed for. So I think at every step of the way, I'm sure we've talked to every investor out there. I would imagine there are probably a few we've missed. But I think along the way, we've managed to line up with the ones that are a real fit for what Mission Bio does and what Mission Bio needs out of our financial partners.
In addition to the money they bring investors, are there any standouts that added more value in terms of helping you build the company?
I think the strategic partners have a lot, and I don't want to underestimate or undervalue their contribution for all the reasons that a strategic partner is there. Way more than just money. These are large, incumbent, existing infrastructures where we can lean on expertise from an entire community that already exists. It's a partner where it's not just a partner at a fund, which is useful, but it's thousands of people behind them who are also working in the space where we can leverage their expertise. So where questions come up along the way, you've got immediate answers. Again, we've had just such tremendous support from strategic investors, which I think has been just so important in leveraging commercial relationships and really getting the product scaled up into market just much faster than we'd be able to without that added expertise along the way.
Were there any resources that helped you guys, like incubators, advisors that came along the way?
Yes. Starting right from day one, I think, Adam, you've really leveraged the QB3 startup in a box, which was, I think, brand new. I think you guys were one of the very first ones, if I remember that correctly. That was an early one. And I know you guys in the lab leveraged a lot of the QB3 resources. I remember QB3 supported with some IP analysis right from the early days as we were spinning out of UCSF. They supported with IP analysis that's continued to serve the company to this day. And it's something that would have been very difficult for a startup to invest in without that level of partnership. And that was a tremendous resource for us. We were and continue to be part of the StartX ecosystem, the Stanford StartX Accelerator, which is truly one of the premier accelerators in the space. And in my view, easily the top accelerator for medtech and life science companies. So we've leveraged just all kinds of resourcing from the outside along the way. And I think in the early days, much to Adam's comment on the valley of death that the NIH and SBIRs are there to support, I think in much the same way, there's a lot of this infrastructure that's there to nurture early stage startups through that valley of death and cross the gap and get them funded. And I think we really leveraged a lot of those resources along the way.
It's funny because it's so long ago now and it seems so trivial, but at the time I remember thinking, well, how do you start a company? I had no idea. I mean, I was a postdoc, then a new professor. So how do you do that? And how do you recruit people that want to start a company with you, recruit your co-founders? And that's where Startup in a Box was really led by a guy named Doug Crawford, who I don't believe is associated with UCSF anymore, but I would talk to him literally on a daily basis about getting advice about this. And he was such a positive thinker and believer, a glass is always half full kind of guy. And he really put his own time and all the resources that were at his disposal to help you do that. In hindsight, there was some lawyers helped found the company, create a cap table, the IP analysis. It's relatively small compared to what Mission Bio has raised and grown into today. But had I not had those resources, there's a good chance I would never have taken that step because I had absolutely no idea how to get started. So for me, that was very eye-opening to have that support.
Doug Crawford is amazing. He's one of my first mentors of sorts when I started in the Berkeley postdoc entrepreneurship program. He's like, I'll help you. He brought the speakers, he funded the program. That's why I'm here today, talk to you guys. Well, we'll slightly shift the gears a bit. Let's go into the culture and diversity of the company. I know, Charlie, you're very proud of diversity you built at the company. Can you talk about that, about the recruiting process, what went into the cultural aspect? How do you build that culture?
Absolutely. We've always been a very mission-oriented company. When we bought into the name Mission Bio, we really bought all in on that one. And it truly is a mission-driven company. And that's central to our culture. We serve customers who are right at the front lines of patient research and patient care. These are some of the smartest people all around the world who spend every minute of every day in service of helping their patients and trying to develop new and better treatments for their patients. And as a company, we've always been very humbled by the mindset that we are serving this community on behalf of the patients that they in turn serve. And that's really built itself into the mission of the company. So over time, as we grew deeper into the oncology space, our mission has become just very simply to help our customers eradicate cancer. I'm sure we'll talk about this, but we've onboarded a new CEO earlier this year, and that culture has continued to evolve under Jan's leadership, but very much built on that foundation that we're there to help our partners ultimately help their patients. And that's driven just a really, really strong culture of people who are dedicated to what we're trying to do on the market in terms of the partners that we serve. And what that means as a company is that the culture has always, from day one, been really strong around enabling that mission. We've hired against that mission. HR is really structured around building into the mission. Everything that we've done up and down the company, really from day one, has been in some way supporting that mission, whether it's interviewing against it, but all hands meetings, keeping people strongly engaged in it. It's always been a real cornerstone of the company. And what that means is we've been able, because of that mission and because of what we do in the space, we've been able to recruit some of the very best people around. The company certainly continues to do so. And it's because you can work anywhere in the world, you can work on any problem if you're the very best around. And we always want the very best on our team, but you would want to work at Mission Bio more than any other company because of our important mission and because of what we're continuing to deliver on the market all the time. So that tight culture has really helped with hiring the strongest and very best team we possibly could. Hand in hand with that, we've always had a strong culture of diversity, both in terms of skill set and in terms of background of people that we hire. And that's always been a very thoughtful and deliberate philosophy from my end and from the managing team, with the mindset that we have a product that's never been built before. It's a complicated product and it's extremely multidisciplinary. We came out of a microfluidics lab, so certainly there are devices and microfluidics, but it's advanced chemistry, it's molecular biology, it's hardware, instrumentation, software is really more and more a lot of what we do. Software and analytics is a huge part of us. Operations, manufacturing to scale that up at the quality that's needed by the customer. Just every scientific discipline, for the most part, is captured under the hood of what Mission Bio does. And in some ways, that's by necessity. We've built an elegant but sophisticated solution to simplify what is very complicated biology and turn it into insight that's meaningful in terms of patient care. And in order to simplify that complication, we provide a technology that itself is fairly sophisticated and to a large extent complicated under the hood in order to make it simple for our partners. And that diversity of disciplines, the diversity of skill set, I think goes hand in hand with diversity of thinking, because we're solving very important problems we have right from day one. And it's really important to have diversity in thinking and openness and ability for everybody to listen to each other in a diverse environment. And that mentality that everybody brings something unique to the table based on your background. And because of that, we're able to build something complicated that's much bigger than what each of us could build on our own. That mindset of diversity has always been baked into the company.
That is as complicated as it gets with all the diverse fields that you mentioned, like everything from biology, microfluidics, engineering, and so on. I think to knit all this together, there must have been very good communication going on to build a really good functional product. What were the steps you took to have that free-flowing communication? What were the issues you found? How did you correct them?
To me, in the early days of the company, we took great care to keep a very open environment. Literally down to the aesthetics of the company. In the early days, we had an open office plan where this multidisciplinary team was able to just be with each other all the time. So I think just an open and communicative style is really important in the early days, especially because everybody comes from different backgrounds, whether it's physics or biology or engineering or from the commercial side. And those different backgrounds are all needed as voices in the early days. So we had a fairly flat structure early on. And again, I'm talking through the days of the grants and the early days of the company. very flat structure. Everybody, if not driving decisions, at least involved in understanding decisions. And with all the information coming from the people who would ultimately need to support those decisions, everybody engaged every step of the way. And that was always an important mindset in the early days of the company, even as we were venture backed and building to early market. Very important to have that openness and transparency across the company.
How do you deal with disagreements?
Disagreements are always hard to move through, but an open and transparent understanding of what's behind decisions is always essential. And an open environment for communication around decisions, I think, is really important. And especially in the early days, this is so important because every single person at the company, again, I'm thinking back to the days when Adam and I were very closely involved in development and technology direction. Every single person at the company has such a critical hand in building it that it's so important to hear from everybody's voice, because everyone is pulling so hard and everyone matters in such a big way that if you're ignoring someone, you're ignoring important information. And you always want to go into a decision with the best information at hand. So the key to disagreements, I think, is to have an open and collaborative discussion, but then an understanding that this is a startup company. It's not an academic environment. Our goal is to build product for the customers who need them and serve, again, always redirect back to our mission, which binds us together. Always our goal is to serve our mission of enabling our customers to cure cancer. And when you keep that as a North Star, again, with that mission kind of binding together everything the company does, it sort of helps bond people around decisions. So hopefully, if you've asked for input from everybody, and especially the people who will then be executing the decision, if they're not making the decision, at least they're giving input that will feed into it. And afterwards, explain why the decision was made. So that full transparency is essential. And if you have an environment like that with everybody understanding and appreciating that everything we do is in service of the mission of the company, there will be disagreements, but everyone will agree to commit to a decision, which is always the most important thing to a startup.
Yeah, I agree. The transparency is super important. I think listen to everybody and telling them why a decision is made is super important. It's a nice culture to build around. Well, Charlie, you've been a success story by every metric, I think. Building a successful team, product, raising money. In the life of a startup, there are always transitions that happen in terms of leadership. It looks like you guys have a new CEO at the company. Walk us through the decision process, like why now and what is the future looking like?
I view the recruitment and onboarding of Jan, who is our new CEO, I really view that in many ways of the success of Mission Bio. And it's really humbled me to see that Mission Bio is at a stage to be able to recruit a world-class leader like Jan to continue carrying the company forward. So after the Series C financing, I think there was strong recognition that Mission Bio was really poised for success on the market, for continued success, and that there was strong resource to back that success and to continue to enable the company to build into the mission that's always driven what we do on the market. And I came to the appreciation that while I was very proud of having built the company to the stage that it was at, I also wanted to make sure that I had the very best leadership around the table to continue carrying the company into its ongoing success. At a startup company, you're always evaluating leadership as you grow to figure out whether it's the right leadership for this stage. And you're always thinking about not what do I need now, but what do I need over the next several years? And you're moving so quickly that you want to kind of think within a several year time window. And it's really important to be introspective across the management team and between the management and the board, and to make sure you've got the right talent around the table at every stage, because the talent in the early stages is not the same as the talent that's needed in the later stages. And it's not that it's better or worse, it's just different. And at every stage, I think it's important for a CEO to also evaluate herself or himself together with the rest of the leadership and with the board, and figure out whether every member of the team is in the right role for the company at the right time. And I think I had an appreciation that I was very proud to build Mission Bio to scale on the market to the impact that we had intended to build to on behalf of our partners that we serve. And that to be able to continue to do that and to really deliver on the mission that we've set out, I thought it would be really beneficial to the company to have someone really experienced at continuing to lead through the markets that Mission Bio is continuing to embark on in NGS and oncology and cell and gene therapy, with just real operational experience scaling quickly through these markets to be able to serve as many customers as we possibly can with the impact that we can deliver for them. So to me, it was a decision of the right leadership in place at the right stage of the company. And I'm just so proud that we were able to recruit someone of Jan's caliber to continue carrying the mission forward.
And I think that's actually, it explains a lot too, how my role has shifted in this company. I mean, strongly stressed what he said. In essence, every company, it goes through kind of a life cycle. You start out embryonic, and at some point you learn how to walk, and at some point run, and at some point you can do something amazing and have huge value. And the reality is that it's very difficult as an individual who's contributing to that whole cycle to really be relevant through the whole cycle. Each of us is just one person. I mean, companies really are, obviously they're comprised of the efforts of many people with many different skill sets. I mean, I became, more or less term irrelevant. I'm not irrelevant. I don't think of myself as irrelevant. I still do a lot. But the reality is that my importance really waned quite a bit after the company spun out of my lab. And at that point, it was really for me to make myself relevant to the company through my value add. And I think that's true for all people that are at the company at all times in the company. And it's likely that if you are very relevant early in the company, like the academic founders, it's going to be challenging for you to stay relevant through, let's say, the IPO, because what matters to the company most at those different stages changes enormously. Initially, it's basic science and a lot of engineering. And at some point, it becomes really about the financials. And each of us is really expert in one little area. Usually for scientists, it's the science. And what that means is that we really know nothing about the areas that could matter at a later stage. It's an inevitable, I think, side effect of the success of a company that has grown beyond its original starting point.
That's a very important point. I think that introspection of like how relevant we are as the company grows is super important for all the founders, especially to think about if they want the company to succeed, I think. There are so many fascinating lessons in this journey that you shared with us, Adam and Charlie. Thank you so much for that. Now to the closing questions. I'm going to ask this to all my guests. So can you share one or two lessons that you learned during this journey that other startups could benefit from? Any mistakes to avoid or any good advice? Adam here from a faculty point of view, who founded a company, and Charlie as a CEO.
Maybe I'll start because I probably have less to say, but I think kind of lesson number one that I think is super important as an academic is to recognize that you're going to be hugely in need of other people and their help. And your prime focus should be to recruit people and give them as much credit so that they really buy in to your idea. You should be so lucky if you can get other people to spend a significant portion of their lives working on your idea. So don't be selfish, share the wealth and the credit, and really try to get the team all on board. And then the other thing I would say, and this is honestly could be a much longer conversation, a whole other podcast, which is what is the role of academia in founding companies? In many ways, I think it's a non-trivial idea because as academics, we think of ourselves as these purists when it comes to science and research, and that it should be totally independent of something as dirty as making money, especially for ourselves. But the fact of the matter is that we're benefiting all of humanity by advancing technology, and academia is one of the largest engines of new technology development in this country. So it's a tightrope as an academic to walk. And I think that each person has a certain amount of comfort with what they're willing to do and what they're not willing to do. But it is personally a role that I find extremely important, that I'm very much committed to. And I think more academics would have a greater impact if they were committed to it too. And if you could just get comfortable with the fact that it is a tightrope, but that it's also hugely important to the advance of technology. And I mean, if you think about it, like, why is the country investing so much money? They take money from people in the form of taxes and they give it to us to do research. Don't we have a responsibility to give back in a really tangible way more than just writing our papers? And I can't think of anything more important toward that than commercialization. So take that aspect of it very seriously and be willing to kind of get your hands dirty in trying to figure out how to make these technologies actually come into existence.
Great lesson, Chris. I'm going to take you up on that offer, Adam. I'm going to bring you back and a few of other faculty members who think alike. I think we'll have a discussion, a nice panel discussion. Charlie.
Two very simple lessons. The first is to always hire the very best people you can at every stage of the company. And the reason I say that is that a startup is inherently a risky venture. It's risky for investors. That's why the angel capital is so expensive. It's risky for entrepreneurs because you're putting time and years of your life into this and you hope it's going to play out. It's a risky venture naturally. The technology is risky, particularly for academic spinouts. There is so much risk everywhere, all over a startup company. There's no sense putting risk in the team. And I say this all the time, but at a startup company, you are naturally building a product for the very first time. No one has ever done it before. And that's the most exciting thing about a startup company. It's something that's never been done. And you have a conviction that it can be created in a way that the market will adopt it. And with that mindset, you have a technology that's never been invented before. Why not find the people who have the very best skill set to be able to do it? And what I look for is people who are not necessarily experts in building our product, but people who are versatile across lots of different technology areas, and especially the interfaces between technologies. Because again, no one's ever built this before, but if you're versatile with the breadth of skill set, and you're able to put on a creative hat and work collaboratively on a team toward aggressive goals, then you will inevitably be able to come together on the team and get the product built. So you'll give it your very best shot if you can hire the very best around. And I think this is especially important for academic spinouts, and especially, I would say, for first-time academic founders, because there's a mindset that I've seen that I don't think I can hire the best. I've just spun out. I'm small, barely paying myself. How could I afford the best? They won't even take my call. And I would completely turn that around. And I'd say that if you have something worth building, if you have something that you're going to invest years of your life in because of the impact that it's going to deliver on the market, you have something that's important for mankind to have built, then why couldn't you hire the very best? Because anyone could come along with that vision. If you've convinced yourself, I mean, certainly if you've convinced investors, you deserve the very best. And with that view, if you're able to convince others to come along, and if you're able to bring the very best along, then you truly do have the very best idea. All of that brings me to my first point. Always hire the very best. Don't settle for less, and build the very best team around that. It's very important for the team to have complementary skill sets and for them to be able to work together. But why not hire the very best into every role? The second piece of advice, very simple, always listen to the market. From day zero, I would say, before you've spun out, from day one, the day you've spun out, and every single day thereafter, listen to what the market is telling you. You're not just building a technology. You're not just taking in government money or investor money to build something interesting. You're building to an important market opportunity. And you always need to be talking to customers. Listen to what they have to tell you about what you should build. And if you're always listening to customers, then you will build something that's important for the market.
I'd like to just say one other thing on that made me think of something, which is that, again, I'm thinking back to my mindset when I first got started in commercialization. It was, honestly, I thought, well, if I could just get the money, it's going to be fine. And I really, in hindsight, have learned that that's just not true. You know, money is there when you're ready to execute on a good idea. At the end of the day, it's the fundamentals of that idea which have to be connected to a strong market and a strong team that matter. You know, if you have that idea, you have the market, you have the team, the money will come. You'll figure out a way, but it shouldn't be the be all end all. I think for a lot of academic founders, at least for me at that point in my career, it was, and that is just not the case. I've learned it through experience.
Great points. Great points. Hire the best team and listen to the market, and money will come. And my second question, can you name one or two people that really, really played a significant role in your startup journey?
I would say for me, the early mentorship that I got, I would call out, I guess, two people. The one that really set me on this path was my postdoc advisor, Dave Waits, who's just a phenomenon of a human being and scientist. And he really showed me that you really can come up with an idea and commercialize it. And I don't think that simple idea had ever occurred to me before that. And I actually saw how it was done, and I realized that it was something I could do. And then beyond that, I would say was the early mentorship I got from Doug Crawford to actually create the company and take those steps without getting scared too early on and just never taking the plunge was super important.
One of the most important people to Mission Bio in the early days, and remains so right to today, is a physician researcher at the Hutchinson Cancer Center named Jerry Radich. And this goes in line with my earlier comment to listen closely to the market. Jerry is someone who, at the time when we first met with him in the early days of the company, he was on the steering committee at the NIH for evaluating new technologies for leukemia, for biomarkers and development for leukemia. He's someone who's dedicated his career to making a difference for leukemia patients, both as a doctor one-on-one with his patients, but also as a scientist. He's got a very well-established and very well-regarded lab at the Hutchinson Cancer Center, which was actually the pioneer in single-cell genomics tools applied to leukemia, because he built out the first set of assays that demonstrated that single-cell analysis can yield important discoveries and has important implications for disease progression for leukemia. So in many ways, a lot of the work that Mission Bio does from a market perspective stands on the shoulders of what Jerry and his colleagues did much earlier than Mission Bio. In the early days of the company, as we knew that single-cell DNA was going to be the first product to build, we recognized that RNA was taking hold and becoming mainstream, and that DNA was inevitably going to be the next analyte to take single-cell into the clinic. We were canvassing thought leaders, and Jerry really was, because of the early work he did in the space, he was absolutely instrumental in guiding our technology to where it was in the first version on the market. And since then, he's continued to be an important partner to the company. But if I could identify one person who came out of the ecosystem and really steered Mission Bio in a solid direction, Jerry has been absolutely key to the company. The other one I would point to is actually, I'd say, a collection of a few people. I've always brought the mindset that I like to hire the people and staff in the management around me who I learn from, the people whose experiences that I can draw on, who really make me a better leader and a better entrepreneur just because of who they are. And again, I think, privileged to be able to hire some of the very most experienced people around because of the vision of what we were building. And some of the early folks who came into the company, we have just top people now, but some of the early folks like Keith Jones heading up product development for the first product, Pat Brooks heading up sales for the very first commercial push. I think of like Paul Billings on the board, who is our early board member steering us in a clinical direction. These are folks who are just some of the most experienced people around, who just being around them made it worth it for me to be building the company with them because of how much learning I got just from being partnered with them and being side by side with them. Those are some of the really key people that I think of, at least early on, helping get the company launched.
Great. This is a fascinating story. Adam and Charlie, you've been very generous with your time and sharing the story of Mission Bio. I wish you both the very best and Mission Bio a lot of success. Thank you so much.
Thank you for having us. It's been a lot of fun.
Thanks, Naresh. This was fun to do. Adam, this was a lot of fun. I feel like I've got a little more insight into how you think. It was fun to talk to you about it.
Yeah, it's funny. It's like when you go to couples counseling or something, you learn about each other. Even though you've been in a relationship for decades, you learn a lot more about each other for the first time.
I'm glad to be the facilitator. Thank you so much.
Thank you for listening to this episode of the Lab to Startup podcast. You can find links to the resources and programs mentioned in these episodes, connect with Naresh, or subscribe to this show at labtostartup.com.